in many spaces in your textbook, they'll call the plasmid an R plasmid. All that means is it's a plasmid that has resistance genes. The R is for resistance. So that would be a resistance plasmid. Carries a gene for resistance to antibiotics. But a plasmid is an extra set of genes, and one of the genes on the plasmid might be a gene for resistance, might be a gene for a toxin, it might be a gene for a special enzyme or protein, and one that's definitely on the plasmid is the gene for the pilus. So when a bacterial cell receives a copy of the plasmid, it becomes F+. It has a pilus now. It has a gene for pilus. Remember what a capsule is, or a glycocalyx, or a slime layer? You might get any of those terms. Capsule, glycocalyx, or slime layer. You might see it in any of those terms. What does that help the bacteria do? What? Biofilm, form a biofilm? Good. And why are we concerned with bacteria that form biofilms? They're more antibiotic resistant. The biofilm helps them be antibiotic resistant. And that question also comes up on your third test, I think it is. Maybe it's this test. It comes up on at least one of the tests, I remember. We've talked about biofilm. Biofilm, they have quorum sensing the bacteria in the biofilm, so they kind of like do chemical communication and form that biofilm. And one of the things that's hard about bacteria with a biofilm is the antibiotics can't penetrate the biofilm, so it's more antibiotic resistant. Also, we talked about another function of capsules or glycocalyx or slime layer. It helps it escape phagocytosis. It can hide its antigens from the phagocytes, and also it's harder for phagocytes to essentially engulf it because it's kind of gooey and it'll slip away. And also we talked about fimbria. Remember, fimbria are little fingers for hanging on. What's a gram-negative diplococcus with fimbria? Gonorrhea. Good. Neisseria gonorrhea. Hanging on inside your urethra with its little fingers. And also, it'll hang on to the sperm cell. So when the sperm cells come out the urethra upon ejaculation, the gonorrhea bacteria grab onto it like they're riding a rocket ship up into the womb, so they can transfer themselves into the female. Life just gets weirder and weirder, doesn't it? I mean, that's a piece of information we didn't have when I went to college. Okay, and also, don't forget, sometimes you'll get mix and match, like which is the mismatch, or which is the correct match, or whatever, which ones are bacterial, which ones are protozoal. Protozoa can have pseudopodia if they're amoebas. They can have flagella if they're phylum Mastigopterans. They can have cilia if they're ciliata or ciliophora. Well, I forget which name I used. Ciliophora, I think. Okay, so eukaryote structures, they didn't go away. There'll be some of your choices. So you have to remember which ones are eukaryotic structures and which ones are prokaryotic structures. And also, okay, got that one. All right. Do you remember different kinds of bacterial cell walls? You have a gram-negative cell wall. You have a gram-positive cell wall. Then you have an acid-fast cell wall. What's different about the acid-fast cell wall? Glycolic acid waxes in the wall. Good. And then you have the genus that has the acid-fast cell wall. What's that? Mycobacterium. Good. Name two diseases. Good. And what's the one that has no cell wall?
Non-acid.
Mycoplasma. So you have Mycobacterium, has a cell wall, like a thick gram-positive cell wall, except that it also has the mycolic acid waxes in it. That's what makes it so tough to treat. Those waxes take a long time, first of all, for the bacterium to make them. And in fact, that'll come up on a test question. The mycolic acid waxes take a lot of time and energy for a bacterial cell to synthesize. That's why mycobacterium grows so slow. The slow grower can take up to 2 weeks to show up on a Petri plate. And its life cycle is, I forget how many days. Something like 5 days or something, but it takes a long time for them to show up. They're slow growers because of those mycolic acid waxes. But the energy they invested and the time they invested in making those mycolic acids really pays off, because antibiotics can't target the bacteria, can't get past those waxes. So it helps it survive, makes it more antibiotic resistant. So you had mycobacterium, and then you have mycoplasma. Let the plasma part remind you that the plasma membrane is the outer boundary. And what does mycoplasma cause? Atypical pneumonia. What's a common name? Walking pneumonia. Because you don't get the typical symptoms. You're up and walking around. You're not down and sick in bed. Okay. Mycoplasma has sterols, like cholesterol, in the plasma membrane. It makes the plasma membrane a little firmer, a little more protective, because they don't have a cell wall, so they have to toughen up that plasma membrane. So they do it by having sterols, like cholesterol, in there. So, let's see. What kind of cell would have a cellulose cell wall? Who?
Plants.
Plants. What kind of an organic molecule is cellulose? Good, it's a polysaccharide. Alrighty. Oh, well, here's a bacterial structure I almost forgot about. Endospores. What are endospores used for? By the bacteria. What are they used for? Survival, right. Survival in harsh conditions. The bacteria is going to die. But in order for the species to go on, the endospore is the dormant stage. So it can re-germinate back into the same bacterial cell. Theoretically, it's the same bacterial cell that comes back, kind of like the phoenix. Crashes, burns, comes back from the flames. It's not reproduction, because one cell would make 2 cells. That's reproduction. This is one cell dies, its endospore is left. Endospore gets into a new host or better conditions in the soil, regerminates back into the same genetically identical cell. Okie dokie. And the endospore contains a copy of the genome, a copy of the nucleoid, the genome and the nucleoid, same thing, copy of the genome, cytosol or cytoplasmic fluid, or whatever you want to call it, some ribosomes, some plasma membrane, and then the endospore coat is made up of peptidoglycan. It also contains hard salts and some tough proteins. And what's the name of the substance that you're going to yell out the car window next time you have road rage that makes it heat resistant?
Dipicholinic acid.
Good, dipicholinic acid, right. So if you're pissed while driving, one time years ago, I'm much more mellow than I used to be, at least I think I am, but I usually drive alone, you know. When you drive alone, you're like singing to the music, or you're cursing out the other drivers, like, you fucking douchebag, go, you know. So one time I was driving with this boyfriend in the car, and he was in the passenger seat, and he's like, oh my God, we could never have kids because they would learn all this language driving in the car with you. Because the whole time I'm driving, I'm like, you fucking douchebag. Find the gas pedal, you know? And I'm cursing up a storm. So when I drive, I curse a lot at the other drivers because they're so fucking dumb. Oh my God. I'm like, go, you moron. Go. I hate that shit in Florida. What is it with drivers in fucking Florida? Oh my God. These people would not survive in New York City, which is where I learned to drive, by the way. In New York City, if you're not already halfway into the intersection when the light turns green, people are going to jump out of their cars and beat the living crap out of you. You know, it's like, go. It's 25 cars have to get through this intersection in 5 seconds. Don't fuck around. medicated, you know? Which actually, that's the only time I can drive with them is when I'm medicated. Yeah, anyway, okay, so we got that. All right. So for questions, you're going to get choices of answers. So, like for example, you get choice: nicotinamic acid, mycolic acid, tannic acid, tecoic acid, lipoteichoic acid. So you're going to get questions where you have your different substances, and they all sound amino acid. You know, the nucleic acid. You're like, I don't know which acid it is. You got to know which acid it is. Tannic acid is the mordant for the flagella stain. Remember from your lab quiz? Mycolic acid is the wax and mycobacterium cell walls, acid-fast cell wall. Dipicolinic acid is the acid that makes endospore coats heat resistant. Tecoic acid and lipoteichoic acid are special molecules in the thick gram-positive cell wall. Did I miss one?
I don't know.
I don't know if I missed any acids, but so crap like that, you gotta somehow get it straight to the brain. So tests aren't really hard. There's just a lot of information,
No.
especially in a 6-week class. All right, tell me about bacterial plasma membranes and eukaryotic cell plasma membranes. How are they similar? What's it made up of? Phospholipids. It's a phospholipid bilayer, and it's got proteins interspersed. The reason I stress proteins interspersed is because when we do metabolism, which is our very next chapter, chapter 5, we'll be talking about the electron transport chain, which are some of the proteins in the plasma membrane. Golden's like, oh God, I can sleep through that lecture too, because he had it already. So you guys are going to have to learn